Okay, so I'm going to give you some empirical results about object organization that I've been doing that start to get at some of these answers. So the first question is, what do we think are candidate dimensions that are going to have a large-scale spatial organization for objects? In other words, what do you think the produce sections of object representations are? Yeah? Faces. Faces gets its own section. We kind of know that, but it's true. That would be a good one. What else? Other ideas? Weather. Interesting. Like, weather and objects don't go together. Think of objects. So instead of thinking of food, think of the set of all objects. Weather's a tricky one, but I like that you could have like objects and then atmospheric phenomena might be in their own section. Okay, yeah. Words, yeah, good. That the text strings might have a different status than pictures of objects. That actually turns out to be true, yes? Motion. Sneaky, you probably knew that already, but yes, indeed. But that's a general property. Tell me how it relates to objects. What do you mean objects that move versus objects that don't move?
Yeah, objects that move inside.
Ooh, see how I fed you that one? That's good. So you might imagine there's like a major joint between the kind of objects that are like cars and vehicles and hot air balloons, and those are really separate from things like bookcases and desks and things that are really stationary. Yeah, that would be a good division, perhaps. On that line, I'm going to push you a little farther. Why is that a good division? Objects that move versus objects that not. Is it helpful for input, output? Why might we want a system that groups all the things that move versus all the things that don't move together? I think so too. I actually think you could argue either way, like these ones are all moving, so I'll just shunt them to the same place and then figure out what they are versus, but why, so why output? Why would that be useful?
I'm not sure.
Yeah, I'll make up something. You might need to orient to them differently. When I see a vehicle, I know that's something that's in the moving part, and so I can have really quick readout from that region to go, oh, I need to orient differently, whereas I don't have to necessarily respond really quickly to a bookshelf. Like, okay. So that might be one reason why you might want to group moving things versus non-moving things.
Real versus imagined.
Ooh, tell me more.
So like there's, the objects you come into in your everyday life versus like objects you're reading about in a story.
Like
or
dinosaurs and unicorns.
Yeah, dinosaurs, even like planets and galaxies.
Planets, microbes. Yeah. Actually, someone has asked me that, and I have not looked at that yet. So that's an interesting one that could be. Yeah.
The really emotion on the animate versus inanimate.
Mm-hmm.
Is there one for scale or
Tell me what you mean by that.
microbes versus?
Excellent. On that note, I will transition to exactly what we found so far, because the first, most well-known distinction that turns out to organize a lot of our object knowledge is between animate and inanimate objects. So we've actually known about this distinction for a while. Patients can have selective brain lesions that impair their ability to recognize animals, but they're perfectly fine at recognizing objects. And you can get the double dissociation, which suggests there's localized knowledge for animals versus objects. Yeah?
What about inanimate, like a stuffed animal?
Yes, good question. I don't know the answer, but it's so low-lying fruit. And that gets at a really important question, which is, this cortex, is it about things that are animate in like the true sense, like this is a being with a life, versus inanimate, or is it about things that look animate? Like you can tell that's a bunny, even though it's not a live bunny, it's a stuffed animal bunny. And so maybe animate, so if it's about the shapes of animals, then a stuffed animal bunny should go pattern with a real bunny. But if it's about the essence of what the object is, then a stuffed animal bunny should pattern with inanimate objects and not with animate objects. It turns out, I think, there's evidence for both, so it's a little bit of both, but mostly stuffed animals pattern with animals. which tells you that this division is not necessarily about the color, I know that that's alive and that's not, but more like these are things that look alive or could be alive or have shapes of things that look alive, and these don't. So that's, I jumped down to a really simple point, but that sort of gets at what's the nature of this representation really? When we're dividing things by animate and inanimate, is it about shapes of animals versus the shapes of objects? Is it about some essence? These are really alive and those are really not. It seems to be more about the shapes of animals. Yeah?
So have you tried experiments with like objects that were unfamiliar to the subjects?
Yeah.
So like you took something where they had like completely no idea whether it was animate or inanimate?
Yeah. You want to know what's really interesting about, so I've explored this a little bit. Let's see if I can, let's do a little demo. Let's just see. I'm going to draw a completely novel object. This could go poorly, but we'll see how it goes. I tried. It's supposed to be a 3D object. Okay. When you see that, what does it look like? Nothing? Anything? Give me, if you had to give me a guess. What? Yeah.
A tree trunk?
A tree trunk. Good. Broccoli. Yeah. A wand. Good.
I would say a piece of toy, like maybe like a Lego part or something.
Exactly, like a small little Lego part, right. Okay, so one of the things that I have discovered trying to use novel objects is that when you present some novel shape information through our visual system, your visual system cannot help but go, I don't know what it is, but it looks like broccoli, or I don't know what it is, but maybe it's a Lego toy thing. Like, you can't help but ascribe meaning to it. And it turns out that actually matters. Like, there's almost no such thing as a novel object, because as soon as you see something new, you're always interpreting it with respect to the visual knowledge that you already have there, and the shape features that you're used to seeing that are associated with some kinds of objects versus another. So actually getting the pattern for true novel objects turns out to be empirically just really challenging, because it's really hard to take meaning out of shape, because we just can't help but see parts of shape as having parts of features, and then it becomes really hard. Maybe not impossible, but definitely more challenging than I thought when I started to do this and realized I couldn't. Yeah, you and then you, yeah.
Do you think that maybe babies would see objects as novel? Would their recognition?
That's a really great question. So you guys are good. So we're already starting to get into the core, like to really understand the system. Well, you got to train it with new stuff. Oh shoot, our visual system's already trained. Now you've got to get really early babies in and see what their priors are. So at the very end of the talk, I'm actually going to tell you about some stuff that's not my work, that people have been doing, training baby monkeys, giving them really intensive exposure to certain kinds of things to see what happens in the brain. So let's come back to that. I mean, it's going to be all the way at the very end, but yes, you guys are really along the right questions, which is like, to what extent is this about visual experience and familiarity driving the major divisions, or to what extent are those divisions built in? So in babies, we know that this division between animate and inanimate is really early. Like, really young babies orient more to things that move like animate things or have animate shapes, like really early. So there's some argument that that kind of distinction, that we may be set up to sort of expect that there's two kinds of things that are agents and have beliefs and desires, and things that are objects that we manipulate with. So I think this one might be pretty primitive. But yeah, you need to scan babies or baby monkeys, or these are, yes, go. Wait, no, you do, and then you. Okay, yes.
You're gonna go ahead.
No, I had the questions.
Okay.
Same.
So, what would be a possible answer to how you would just ascribe possible objects to the novel depiction? And does the visual cortex like recruit different sensory areas attached to like discovery with this novel object? To like how it branches out? Does that make sense?
Yeah, you're sort of going, how on earth is it the case that we ascribe some meaning to that?
Right, I mean, like, why did you just like, how did he just say broccoli, and
Right.
why did you just say just like some toy or like?
Right. So wouldn't it be interesting if I scanned your brain when you saw we'll just say you saw broccoli, see an actual picture of broccoli and a tree trunk, and your brain when you're seeing an actual piece of broccoli and a tree trunk. And then I'm going to scan both your brains when you see this. The question is, does this pattern, when you're seeing this, look more like broccoli than tree trunk? When you're seeing it, does it look more like broccoli than tree trunk? Because that would suggest that the way you construe it actually changes the brain pattern. Or, since you're both staring at the same thing, do you get the same brain pattern? Unknown. We could do that experiment. Isn't this great? Part of this is just like, there's just so many questions to explore on this that almost all of these we could go try and scan. So these are good questions. I don't think that one's known yet. Yeah.
Are you using MVPA, and is there enough information for you to be able to do a single object like broccoli?
Great questions. I'm going to hold off on MVPA because I'm going to show you some of the methods as we go. And then, yes, but to get single items, you can. It turns out that's not about the analysis method, but really how long you're willing to stick someone in the scanner for. It turns out you want to get a really reliable brain pattern to broccoli. They've got to see broccoli a bunch of times, and you can get a stable pattern. And really, there's usually a trade-off between how bored the subject is and how much power you want for that particular image of broccoli. So I'm going to talk about a study that I do that is related to single item responses again at the end. And I have to move on because holy cow, it's already 20 minutes. Okay, so animate and fundamental division, we've known about it from babies developmentally. Babies are centered of this division. And patients with lesions have deficits in one category or the other. If you scan human participants and look at regions that are more responsive to animals versus objects, or objects versus animals, you find that they cluster. So here we're looking at the ventral surface. This is a set of, sort of a patch of cortex, and all of this cortex responds more to objects than animals. And right next to it is the opposite preference. All of this cortex responds more to animals than objects. And we did this, but we took a slightly wider view with more power. And what we actually found is that you see these alternating in the cortex. So this whole swath likes objects more than animals, objects, and objects. So this is one dimension that generally provides large-scale groupings of different kinds of objects, animate versus inanimate. All right, scale you mentioned, going way back. This is what I lucked into in grad school, really, is the distinction between big and small objects. Why is this an interesting dimension? Well, small objects you can hold in your hand. They're very manipulable. They have certain kinds of functions. Big objects, they support your weight. You can navigate with respect to them. They have boxier shapes because they have to withstand gravity. So there's lots of reasons, both input and output reasons, why you might separate small objects and big objects. Like those might be two sections in your IT grocery store. And it turns out they are. So if you look, is there anywhere in the brain that's more responsive to big objects versus small objects? You can see there's this swath of cortex here that's more responsive to big objects than small objects. This whole side is more responsive to small objects than big. And then you've got this other sort of big swath up there. So size is another dimension that sort of separates out the space of object categories. So that's two. As far as I know, these are maybe the only two we know. Like faces are interesting because that's sort of like one scale down within animate. There's like faces and bodies. So I think that's it's a pretty large scale organization, but anyway, so this is sort of where we're at. There's probably more, but yeah.
Do you know if there's been any studies with regards to like a doll face and
Yes.
human face?
You're good. These are great questions. Doll faces activate face areas, but there's arguments they activate them less, and or that you can tell apart doll faces and real faces with MVPA, suggesting maybe there's something in the main response that's telling you about faceness, but maybe there's something in a finer grained response that's telling you about, like, is this a real person versus but I think that there's maybe one or two, Talia Wheatley does those at Dartmouth. I think there's some evidence that you can tell apart doll faces and real faces, but it's subtle. It's not like doll faces are down here and real faces are up here. It's more like the more they look similar, the more this cortex seems to be driven by, yeah, good.
I want to ask,
Yeah.
So the one on the left is partially inflated, and the one on the right is
Fully inflated, yeah, sorry.
But it seems like there's similarity between the maps on either side.
Like really similar, right? Oh, good. That was what I did for my postdoc, which is I found this in grad school, and then people knew this, or well, they knew part of it, and I was like, wait a minute, I got the same maps. But if you think about it intuitively, take your objects, you can divide them by animals and objects, and orthogonally, you can divide those by big and small. So you can have small animals and small objects and big objects. So we should see 4 clusters, right? So if you think about how you would put this onto your cortex, if big to small goes this way, then you should put animals here, and you should put objects here, and so then you can get your 4 different kinds of clusters in 4 different regions along the cortex. Intuitive prediction about how you take the space of objects and you divide them into your produce sections. But as you pointed out, both these dimensions, animacy and size, have the same medial to lateral organization. So this has to be wrong. So something has to give, and that's what we figured out next. So in order to explore this, we did a fMRI experiment where we scanned people viewing small animals, small objects, big animals, big objects. So 4 conditions now instead of 2, really leveling up. And we replicated sort of the main results. So if you just look at all small things versus all big things, you get that. If you look at all animals versus all objects, you get that. Our conundrum is still intact. How the heck are these both true at the same time? What's going on? And so really you need a way to compare all 4 conditions at the same time on a 2-dimensional surface. So part of the challenge of this is actually thinking, how do you analyze the data that's going to let the structure show? Like how are we going to figure out the answer to this? It took me probably 8 months just trying different things till I figured out this surprisingly simple answer that's maybe obvious in retrospect. But what I did was define a bunch of regions of interest along the main axis of variation. So you can see there's maybe like 30 different ROIs. And then I can look in each one at how much each condition activates them. And since I've changed the cortex from a 2-dimensional surface to a 1-dimensional surface, now I can see them all at the same time. What do I mean by that? This swath of cortex, I'm now plotting here. So, and then on the y-axis, I'm plotting relative beta, or relative activation. So, they average to 0, and the one that drives the cortex the most is going to be highest. So let me walk you through this. If we start at this part of cortex here, the most medial, hippocampal cortex, it's simultaneously more active to big than small, and objects versus animals. What the heck is going on? Well, you can see that here. In blue, it's most active to big objects. It's intermediately active to small objects, and it's least active to animals in general. So they're simultaneously big versus small, and objects versus animals happening at the same time. One gyrus over, the pattern completely flips. So now in the fusiform, it's most active to animals and less active to small and then big objects. One gyrus over, there's actually a 3rd pattern. It likes small objects the most, animals intermediate, and big objects less. So there's really sort of 3 different peaks in this space across the ventral surface. And then what we found is if you keep going around to the lateral surface, you get a really nice mirrored organization. So fusiform and lateral occipital have the same pattern. And this area here, transverse occipital, has the same pattern as hippocampal cortex. So what we basically see is there's this mirrored organization that goes all the way around the ventral and lateral surfaces. At the center of the map, there is an area that likes small objects the most. On either side, there's a section for animals. And on either side, there's a section for big objects. The second thing to notice that we did not expect is that the distinction between big and small things only applies to objects. So if you look at big animals and small animals, the pink and purple lines, nowhere is there a difference. It turns out that the brain doesn't have a section for small objects. The brain doesn't have a section for small animals and a different section for big animals. Those are just all mixed together. And what this means is there's sort of 3 different sections, so we can visualize that on the brain like so. So what we see is there's basically 2 long, elongated zones of object preferences, which I think is kind of cool because back here is primary visual cortex, and you almost get these spokes or aisles of a grocery store, if you will, that basically route you from very early representations along object selective cortex, along key divisions of objects. So right, cortical aisles, maybe. And what's also interesting is these are partially inflated brains, but less inflated. You actually find that these functional divisions follow the structural divisions, the folds. So, this is the fusiform gyrus. So there's a really nice sort of anatomical correlate to this functional organization, which we know is true in early primary areas, and to start to see sort of structural functional correspondences in these higher level areas is pretty exciting because it suggests maybe we're getting to the right track. Yes?
I know like V1 is supposed to have like replicated in V2 and V3, so there's like pattern.
Yeah.
Do you see like that sort of thing in this too?
Yeah. That's a good question. So, how to answer that? In general, the part of the brain that you can't see on the inside, it goes V1, V2, V3, V4. So these kind of like extend off of V4. So if you think of it like the earliest processing, earliest information goes into V1, and you get a little more abstract and a little more extract. By the time you're out to V3, V4, now you're starting to pick up on object divisions, and these sort of run orthogonal to that. Like you've routed your object information for big objects here, and then it goes on. So yeah, so if anything, this duplication that you see on the lateral surface and the ventral surface, it's not the same as V1, V2, V3. These kind of, let me just draw it. This will be easier.
Kind of like some face replication areas?
Yeah, so it sort of looks like this. If this is V1, V2, V3, these kind of run off of it like this. Big objects, animals, and then small objects. So it's almost like your information comes in, and by the time you got here, it's now routed into the right channels, the right content divisions. So now so if anything, there's like a duplication here and a duplication here. If you look at the organization of V1 and V2 and V3, this tends to correspond to the upper visual field, and this to the lower visual field. So it's almost like you've got a copy of the information based on stuff that came in from like down here where your hands are, and stuff that came in from up here, like that's far away and on the horizon. The stuff in the lower visual field is also motion sensitive. Going back to your point, this part seems to be way more motion sensitive. So there's all sorts of, again, we're starting to get into there's many similarities, many ways of grouping it. And this is in the weeds. You're asking a good question, like I don't even know the answer to this, but I can tell you these are the kinds of patterns we're seeing, but how they all go together, still working that out. Did that sort of answer your question about the duplication of V1, V2? Yeah, good.
Is the non-categorization of small and big animals maybe due to us not being sort of in the how do you say prey?
We don't have that.
A big animal that's going to eat us, so we just like.
Yeah, I think so. That's a good question. You're sort of getting into like the needs of that. Like, if big animals and small, like if big animals were really dangerous and small animals were not, then that would be a useful divider because you might need to respond differently. But spiders and lions, both dangerous, whereas like, hamsters and give me a big horses, not dangerous. Well, horses terrifying, but anyway, you get the idea. So I think your intuitions start to go the same way as mine. Is like, hey, maybe that distinction is not one that's not functionally relevant for the system, and so we haven't sort of made that large scale division. In the grocery store metaphor, that's how you would interpret it. So yeah, that's how I usually explain the fact. I wouldn't have predicted it ahead of time, but now we're starting to see, well, let me see if behaviorally we can see evidence that we're really sensitive to big and small inanimate object differences, but not that sensitive to big and small animal differences. So we're just doing that now. Yeah?
Also, I was going to ask, like, what range of like difference in size did you show to the subjects? Because you have like mountains versus stones, but animals you don't really have, like you don't have dinos or anything.
That's right. That's a good question. So 2 parts to that, which is, hey, is there even more continuum? Like, are these, is here something like chairs and here something like mountains? Like, is there an even finer grained map of size? It's hard to tell. I think we need finer measures. There might be, there might not be. Some subjects that I scanned seem to have that, some didn't, so I'm not entirely sure if there's an even finer grained map. What I've mostly found is that there's sort of like a functional division between things that support you, like a chair, and things that are handheld. That's usually where the small, big border is. For animals, it's true. In this set, I tried to sort of roughly match the sizes of things so that I didn't run into that problem. Like, there are no mountains. Like, there's bookshelves and trucks and things. But so I tried to match the range. But I don't think that actually matters in the end. I think there's maybe, I don't think there's a really smooth map of size. I think it's more complicated than that. It's not a great answer, but there it is. It's a good question. I just don't have the answer yet. This sort of gets to the question about planets and microbes, right? Like, are they at the continuum of these, or are they somewhere else because those aren't things we see? And that's also unknown. Okay.
What?
It's also true for most animals. If you compare a shark to a whale, they might be at the same scale in real life.
But
your representation of whale is really not as...
It's much more abstract, right? It's not necessarily like you...
see every day.
That's right. So I think that's a great question. That's right. Now you're getting to the question of, to what extent are these driven by like walking around in the world, and I get a sense of how big chairs are and cars are and pencils are because I use them, versus more abstract knowledge. Like, we know whales are big, but actually they're like really big, and it's actually even hard to conceive of how big they can be. So in that sense, maybe size is just really not even a everyday experiential factor for animals. So maybe people who grew up on a farm might have a difference there because they see a lot of small animals and big animals. So these are, that would be sort of how much is our visual experience with things in the world driving this organization versus how much is it something more abstract. You can test people with visual experience. You can test people that are blind. They know the sizes of things, but they've never experienced them in the same way. So these are ways to get at that kind of question, like how much is this based on bodily, visual experience in the world. Okay, to your question really quickly, there are face and body regions. So face regions in red, body regions in blue, scene preferring regions in green. This is sort of what we knew going in. And we call it a mosaic because it looks kind of patchy, like raisins in oatmeal, like we just didn't quite understand how they all got organized together. But thinking about where these fall with respect to these broader animacy by size divisions, you actually can see that these fall really nicely along those spokes. So here's your scene region along this sort of big object, maybe navigationally relevant kind of zone of cortex. Faces and bodies fall nicely along the animal line. And so that means that these highly selective regions are kind of meaningfully related to things around them, which is good. So we're starting to get a sense of what the right organization and structure is. Okay, so we started with this question of how do these things map together, and we put in 2 orthogonal dimensions, expecting to see that would map really nicely onto a 2-dimensional surface, but it didn't. Instead, we got this kind of interesting mirrored tripartite organization. We have sort of a copy on the lateral surface and the ventral surface, and we only see this 3-way division rather than the 4-way division we were expecting. And that mostly took discovering ways of visualizing the data to see what was true. It just took an interesting way of plotting it to try and understand what the structure is. A lot of fMRI research, as a caveat or as an aside, is about finding the right analysis or the right way to visualize the data to really get the structure out of it, because it's so high dimensional. You have all these numbers, and most of it is sort of like inventing analyses and inventing visualization techniques that make the data sing. I'm going to skip that because we talked about it.